I will spend the first, actually basically 90% of my talk on activation, reactivation, purging strategies, and then at the end sort of try to introduce what some other people are doing, which includes what Shelby has talked about in terms of zinc and tau nucleases and obviously other gene therapeutic approaches are modifying the stem cells so that they are no longer receptive for HIV, as the Berlin patient demonstrates. And there are actually many things that you can think about interfering beside delta 32 or CCR5 delta 32 if you actually undergo the idea of gene therapy. It's not exactly what one hopes to be able to do with 30 million people in Africa, but it's obviously something that may open new avenues. So basically what I want to cover a little bit is how the infection, because to think about this, you have to really think about how the infection occurs in the first place, because that influences where the virus integrates and where it actually replicates or remains latent, or at least goes latent. And then basically studies of transcriptional latency at least, or proviral latency, and where the reservoir actually might be, and then giving all this information, then synthesize some sort of strategy as to how you'd like to proceed in people. And what we believe in latency, what happens is the virus doesn't really copy itself very efficiently. And one of the main actors here will be this molecule called P-TEFb, which is called positive transcription elongation factor B, which was actually discovered with the viral protein TAT for the transactivator. And P-TEFb is actually the crux of how HIV replicates. In fact, it's the most P-TEFb sensitive transcription unit that we know right now in biology. So this factor is really key for the virus to be made in the infected cell. And the levels of P-TEFb, you have to understand the levels of P-TEFb and how it's regulated to understand latency. And I'll go a little bit into how P-TEFb itself is regulated, because that will form at least one arm of strategies to get rid of the virus. Obviously, the virus brought other things to biology, and you can contemplate these other restriction factors as gene therapeutic targets for making stem cells resistant to viral infection. But that will be at the end. And the other thing I have to introduce to you is that the long-term repeat of the virus looks like any cellular promoter. And you can exclude all of this stuff that's actually in the box here, and all of the stuff here. You can just concentrate on NF-kappa B, 2 sites, SP1, 3 sites, Tata box, and then that's true for both T cells and macrophages. And the most important thing here for everybody in the field is that in terms of DNA binding, NF-kappa B binds very, very tightly. SP1 binds very, very loosely. So SP1 is very easy to remove from the DNA, and NF-kappa B is very hard to remove from DNA. So when we'll talk about transcriptional interference, the main problem with the virus is that the promoter cannot keep the polymerase on the LTR if it does not have NF-kappa B. So, and the other thing that you have to remember a little bit about the HIV promoter, and this is getting very complicated for many of you, that it also has an RNA element, which is important for its replication. And this RNA element in HIV-1 is a simple TAR. In HIV-2 and SIV is a complicated TAR. This binds to TAT, and TAT also requires P-TEFb to transcribe the viral genome. So NF-kappa B requires P-TEFb, TAT requires P-TEFb. And what you will see in a moment is that these little transcripts that are made in the absence of much P-TEFb or TAT or NF-kappa B are in fact the best molecular markers for latency in the host. And you can detect them in infected cells. So HIV actually does not infect resting T cells. So it has to go into an activated T cell, because the pool of triphosphates, nucleotide triphosphates, is not optimal. There are other things that are missing. Even though it can transfer the nuclear membrane, it needs to have an activated T cell to get into a T cell and infect it. And that really limits the of where the virus goes once it enters the T cell and integrates. And so basically, it integrates into an active lymphocyte. And actually, it integrates into active open chromatin, which is another thing that you have to remember. And basically, it can only replicate and integrate after the cells have been activated, not just because of the triphosphate problem, but also because there's no P-TEFb in a resting lymphocyte. So basically, there's no substrate for HIV to replicate. And there's not enough triphosphates. But the problem is once you activate the cells, then 99% of the cells are going to make virus, and less than 1% probably is going to become latent. And we believe that this latency is not because of chromatin, it's not because of position or epigenetics, it's because it lands in the actively transcribed genes where it's transcriptionally interfered. And so basically all the reservoir that we have to deal with primarily if the virus is intact, if there are no mutations in TAT or TAR or other aspects of the virus, are due to transcription interference, and that is being replicated in these cells during the proliferative renewal, for example, of T memory cells. And the virus just keeps going until a point when basically the gene where it's sitting in is either not transcribed or NF-kappa B gets activated and the virus comes streaming out. But I'll go into those steps in a little bit. So basically the reservoir is in resting, we agree with resting memory T cells. It is in a cell where the virus is not being made, and the cell is replicating to some extent in lymph nodes and lymphoid organs. So at least by proliferation, at least the virus gets transmitted to daughter cells in the cells where it's latent, as well as the pool is constantly replenished by macrophages, which are making low levels of virus. So basically, there's clonal proliferation of effective memory T cells, there's low replication, and there's persistent replication macrophages, all keep the reservoir going. And there's blips during the optimal heart therapy, where even more virus is being seeded into the reservoir. So the reservoir is basically not able to be eradicated during the lifetime of the patient by any therapy that targets the virus. Now, as Silicano has suggested, that HIV can hide in this very long memory CD4 T cells that are insensitive to viral drugs. His estimate at the time was that there are basically 10 to the 5, 10 to the 6 cells. We actually disagree with this number a little bit. Together with Paul Lucio, we think that the level is about 10 to the 8 in rhesus macaques. By studies I'll go into in a moment. So there's basically 100 times more of the latent reservoir. And the new approach is obviously urgently needed to either purge or permanently inactivate these proviruses. Otherwise, the cure for HIV infection would remain out of reach. And so why does HIV integrate into active sites? It's partly because of this molecule, LEGF, partly also recently suggested by TRIM28. And basically, the virus really does go into active chromatin. It does not go into heterochromatin. So if you think about chromatin, it goes into this kind of chromatin. It goes to the chromatin which has major minor grooves, but doesn't go into the chromatin that you will see in a moment as the chromatin condenses. So see now nucleosome is coming, and now you're going to get 7S, 30S particles. And then finally, 2 meters of DNA will be condensed into a few microns that are actually residing in the cell nucleus. And so I don't have to go through all of this film, but this was confirmed, first presented by Rick Bushman, who showed, for example, specific hotspots for viral integration. For example, here is CREB binding protein, then there's another housekeeping gene, FBOX, and then DNA methyltransferase. And you can see these little dots are all for integration site of the virus. So basically, the virus really does integrate into active genes, mostly in introns, and there it is transcriptionally interfered. And because SP1 does not bind tightly, the polymerase just rides over the viral genome, and nothing is being expressed. So no viral protein gets just cleaved out as an intron, and basically the RNA is just wasted in the nucleus. So basically all these other ideas about chromatin localization, all this nuclear export, all these other hypotheses probably are not very important. So we think that basically there are only two things that operate in latency. One is the absence of transcription factors. As I told you before, in the resting lymphocytes, there's no P-TEFb, and transcription interference. Now, Bob Silicano and Warner Green have just published a review in Cold Spring Harbor Reviews, and I would just remind you that it's a little bit wrong. We've actually shown very clearly that when the virus is sitting in the sense direction of a gene that's being transcribed over it, it always terminates in the 5' LTR. So this is a very good polynylation site, as everybody knows from viral replication, because the 3' LTR, when the virus starts here, is a very good polynylation site. So all transcription stops here, if it's in the sense orientation. So basically, it's like a gene trap. If it's in the opposite orientation, there is no collision, actually, because usually this polymerase wins out in every single case when there's no NF-kappa B there, and the polymerase just keeps going and occludes the 3' or the 5' LTR here. So basically, in either situation, in the sense or in the anti-sense orientation, by transcription interference, the virus does not fire. The only difference is here that basically this does not inactivate the upstream gene. This does inactivate the upstream gene. So I told you that there's a marker for all this. And basically, if you stop here, you start here. And the most interesting feature about this is when you start in the 3' LTR, when you add that to the system, you actually transcribed very high levels of this particular transcript. But in both cases, it's marked by short transcripts. And the short transcripts we found already in 1987, because we were able to show that in the absence of TAT, transcription was starting but not elongating. In the presence of TAT, it was starting and elongating along the HIV genome. So basically, if you have no P-TEFb, if you have no TAT in the system, you get short transcripts. And if you have TAT in the system and P-TEFb in the system, you get long transcripts. And so this was nuclear run-on. You can see this by RNA protection. So this is short transcripts, exactly the measure of TAR. This is the long transcript in the presence of TAR. And this marks actually lack of transcription factors as well as transcription interference because of the 3' LTR. So basically what we have here is an RNA polymerase that's going to the HIV promoter or HIV LTR, and she will just stay there. She will just basically stay there making short transcripts forever, okay? And until you have P-TEFb and TAT there, or NF-kappa B, and then she will finally get going, and she will transcribe that. So she finally gets going, and she will actually polydendrylate. She will terminate in a moment, but okay. Well, you don't want to see her terminate, I'm sure. So, okay. So together with a number of laboratories, we were able to then show that this is true for HIV-infected people. And we used an RNA PCR strategy, in which case we used primer pair 1, primer pair 2 to amplify short transcripts, primer pair 1, primer pair 3 to amplify long transcripts. And lo and behold, when we started to look at patients, at zero conversion in the peripheral blood, everybody had short transcripts, but nobody had long transcripts. And that is easily explained because activated lymphocytes don't circulate as long as the immune system is intact. As long as the lymph nodes are intact, as long as the spleen is intact, activated lymphocytes don't circulate. So these are resting lymphocytes that circulate, and they have nothing but short transcripts. So we actually thought we defined latency in 1994 until we were told in 1997 that you couldn't describe latency unless you give people heart. But I don't know. I mean, this is latency. I mean, this is basically no virus is being made except for short transcripts. And this is in a cell which is actually targeted by the virus during activation. And now it's come to a resting state. And so this is for short transcripts either from the 5' or the 3' LTR, depending on whether it's transcription interference or lack of transcription factors. But in every case, we were able to activate the virus to replicate by giving here allogeneic cells, so it's allogeneic effect, within 3 or 2 weeks, and the virus was basically able to infect other cells, was completely infectious. Now, we use the same strategy then to look for progression to AIDS. And when you start looking at longer transcripts, that means the lymph nodes are gone. So when ARC transitions to AIDS, you stop seeing this tremendous separation between short and long transcripts, but now you can see in the periphery long transcripts appear. That means that the immune system is shot, activated cells now circulate, and this person basically was terminal and died a few days after the sample was taken. So, just to remind you again, what we're amplifying is this little piece that occurs independent of P-TEFb and TAT and NF-kappa-B. And this is in the monkey SIV. And together with Paul Lucy, we have started to do initial studies on where the reservoir actually is in monkeys which have been optimally treated with HART. And this was a trial of HDAC inhibitor and prostratin in addition to three nucleoside inhibitors or non-nucleoside inhibitors for RT. And basically, we were able to show by just quantifying short transcripts versus long transcripts that the primary reservoir of SIV in the monkey, at least SIV RT-SHIF, which is human RT, is all in the gut lymphoid tissues. And there's about 8-fold more short transcripts than long transcripts in this particular tissue. Very little in the brain, but this virus does not infect the brain, so it's possible that in the neurotropic SIV there'll be more of a brain reservoir. But all the other lymph nodes were quite full of virus, and all of them short transcripts predominate over long transcripts, including lymph nodes, spleen, liver, and so forth, and very little virus in solid organs. But by this measure, and by measuring of viral DNA in these organs, basically we came to a conclusion that the reservoir was at least 10 to the 8 cells in the infected, optimally treated monkey. And probably it's in the same level in human beings. So it'll be very nice to actually develop an in situ system for SIV because the RNAs are longer. And we hope that maybe Ashley Haas or someone of that expertise would actually use this marker or try to develop this marker for staging and also for looking at the reservoir in humans, where you can do certain biopsies, at least in the gut, for example, to demonstrate that it's very similar to the monkey situation. But anyway, the short and long transcripts, I think, at this time are really the only marker for latency that we know of, and I think would be very useful to develop it as a clinical test, especially when the purging strategies are becoming more clinically useful. So, just to get back a little bit to P-TEFb. So I told you already that P-TEFb is the coactivator of TAT. So here is TAT. It binds to cyclin T CDK9, which is P-TEFb. And this is absolutely essential for HIV replication. And it also binds NF-kappa-B, P65. So P-TEFb is absolutely required to initiate HIV transcription as well as to amplify its replication via the TAT molecule on TAR. And there is a problem. There is a problem with P-TEFb. First, the problem is that it's very low in resting cells, primarily because of translational inhibition of cyclin T1. And even after you make it, after you induce it, basically you induce all these other factors that sequester most of the active P-TEFb into an inactive complex, which is either called a large complex or 7-SK SNRNP. So basically, if you take any cell line that you people work with, for example, Jurkat cell line, HeLa cell lines, 90% of all P-TEFb is in this large inactive complex, and only about 10% or less is in this active complex, which is required for transcription. So here the kinase is dead, the CDK9 kinase, which otherwise phosphorylates all these important targets, is inactive. So there was a paradox that we had initially when we started the studies, and that we knew that there was a drug called HMBA, hexamethylene bisacetamide, which is the strongest differentiation agent known to man. In fact, was occupying Paul Marx for about 15 or 20 years because he thought it would be very useful for treatment of leukemias. But it's also the protein, or at least the the substance that induces something called hexim, hexamethylene bisacetamide induced messenger RNA 1 and 2. And the hexamethylene bisacetamide protein 1 and 2 are the ones that inactivate P-TEFb. So how could HMBA that induces the synthesis of hexim be such a potent activator of HIV transcription and replication, at least in Paula Pitha's hands and then later in our hands? And what we found was really very important for all the strategies of eradication, that the first thing that HMBA does, it completely destroys the large complex. Completely destroys the large complex. All the P-TEFb is released. But within 6 hours, there's so much hexim being made from the released P-TEFb that the whole thing reassembles. And the reason it doesn't overshoot here is because the cell undergo apoptosis and die, because all the P-TEFb has now been removed. At least these are transformed cell lines like Jurkat, HeLa, and so forth. So basically, there's a release of P-TEFb by HMBA very quickly, and then reassembly of the large complex. During this release of P-TEFb, HIV transcription is very potently activated. And the reason it's sustained is also an important fact that I'll get into in a moment. It's because TAT does not bother. It's not important for TAT whether P-TEFb is free or P-TEFb is in the inactive complex, because TAT competes directly with hexim for P-TEFb. So the only thing you have to do is to liberate P-TEFb to make NF-kappa B work, or at least the initial rounds of transcription, and then it will be sustained. And that's the Achilles heel of the virus that I think is very important to know, because it's the only gene in the cell that does not mind that P-TEFb is back in the inactive complex. But I'll get into that in a moment. But there is, so HMBA is no good. HMBA is potent, but you need tremendous amounts, 5 millimolar, 10 millimolar. Paul Marks at Sloan Kettering tried to give it to patients. They could never achieve the dose. It was very toxic. But there is a drug that does exactly the same, and it's called SAHA or vorinostat. And this drug is actually approved in clinical use, FDA approved for the treatment of cutaneous T-cell lymphoma. It's being now used in about 20 or 30 clinical trials for cancer. And it does exactly the same thing. It activates PI3 kinase, transiently activates AKT. AKT then phosphorylates 2 sites on hexim 1. Not important which ones there are. Hexim 1 now no longer binds P-TEFb. P-TEFb is released. P-TEFb now can do its job to phosphorylate RNA polymerase and transcription factor so that they can elongate. And obviously hexim and all this other stuff is also induced. So this transient release leads to an activation of HIV transcription, but then also the fact that the cell becomes totally resting within 6 hours of the release. So there's no T-cell activation, which is also extremely important for this particular discussion. So the next thing we actually were able to demonstrate that, and I'll show you some data on that, is that, okay, so you have transcription interference. So you have an upstream gene. The polymerase goes from the upstream gene. It terminates in the 5' LTR, dissociates. The 3' LTR now fires, makes transcripts which are completely, they have no viral information in them. But if you now activate NF-kappa B, then you can actually block this polymerase, because NF-kappa B does bind so tightly that the RNA, and binds also single-stranded DNA, which is important. So the RNA polymerase now is stalled here, and now the HDAC inhibitors, or SAHA, for example, can start working on the initiating polymerase on the 5' LTR. So you need NF-kappa B release and P-TEFb release, and the combination of the two can overcome transcription interference. Importantly, if you do activate NF-kappa B here, you will not go to global T-cell activation because of what SAHA will do to the P-TEFb cycle and inactivate P-TEFb through hexim and 7-SK SNRNP. So just to summarize this, and this is very important, I think, in terms of contemplating of how one should go about to devise a strategy, drug strategy to get rid of the virus. You will need something to activate NF-kappa B to block transcription interference, as well as to capture initial cyclin T1 for initial rounds of transcription before TAT is being made. So this can occur through PKC agonists, and there are a number of them that have been tried now. I'll show you a small list. And then finally, you will have to use something like SAHA to be able to release P-TEFb from the large complex so that this will go to NF-kappa B and synergize to make TAT. Now, the benefit of this entire scheme is that once TAT is being made, TAT does not need free P-TEFb. TAT actually goes to this RNA, TAT recognizes this RNA, but this RNA that binds hexim, the 7sk RNA, is identical to TAR. In fact, I would actually suggest that the HIV virus stole the entire system from 7sk, because this loop which binds hexim is indistinguishable from TAR, and TAT goes there, competes with hexim for the binding of P-TEFb, and now takes P-TEFb directly to HIV LTR and is able to sustain HIV replication, even though most P-TEFb has been reassembled because of increased amount of hexim into the large complex. So the other thing that we've recently submitted for publication is that PKC to some extent will also have a beneficial effect for the small complex, because PKC will directly phosphorylate hexim on a site, serine 158, which interferes with the binding between hexim and 7sk, because this site is exactly in the RNA binding pocket of hexim, and so basically it will prevent a tremendous reassembly of the large complex. So basically both together will synergize not only on the level of NF-kappa B, but on the level of P-TEFb. So just to show you how important this synergy is, so here you have a Jurkat model of latency, and I can tell you that every single transformed cell line that is used as a model for latency is transcriptionally interfered. No matter what you read, every one of them is in an actively transcribed gene. Every JLET clone ever developed by Verdun and Jordan's lab is an actively transcribed gene and is transcriptionally interfered. And so here you can see that the HDAC, so this is a class 1 HDAC inhibitor, class 2 HDAC inhibitors, and they all liberate P-TEFb, so don't worry about how they work. They don't work by histone acetylation. They work through P-TEFb mechanism. They do almost nothing on their own, even SAHA, when you have transcriptional interference. But once you add TNF-alpha to the system, which activates NF-kappa B, you can see now very robust activation of HIV in this latency model. So that synergism between NF-kappa B activators and P-TEFb release factors then produces a tremendous activation of viral transcription and replication in this cell model of latency. Now, the other thing that I just want to summarize again or to stress again is, even though you have a release of P-TEFb by all these HDAC inhibitors, because the hexim synthesis is increased by free P-TEFb, you will inactivate enough P-TEFb so the cells will not go into, so the cells will not be activated or will not basically cause toxic shock syndrome, which is one thing that you worry about in terms of global T-cell activation. So the cells are basically resting or even super resting by this particular approach because of the hexim cycle. So just to summarize, I think that basically there are a few summary slides just so that basically I make this point, that basically you need some sort of NF-kappa B agonist as well as some sort of P-TEFb release factor. And the nice thing about PKC agonists also, they increase the synthesis of hexim, so of P-TEFb. So basically cyclin T1 synthesis actually increase, and then basically we hope that the immune system as well as heart will take care of the virus that is being released by this activation therapy. And I know that Silicano just recently published that maybe the CTL response may not be robust enough after very long treatment of heart, but it's possible that the cycles of release by this particular strategy would in fact induce CTL responses at the same time, so that maybe one doesn't have to boost those in parallel to clear the virus after you activated its replication. So what do we have now? We have, in terms of drugs, we obviously have in this particular PKC agonist, we have TNF-alpha, obviously, prostratin, bryostratin, and ingenol. Ingenol is kind of interesting. Renato Aguiar here in Rio is working on this. It's an approved drug, and it's used for treatment of actinic keratosis in humans. So it's a very potent PKC agonist, and maybe better than Braustatin, which is extremely expensive and very difficult to manufacture. In terms of release factors for P-TEFb, as I mentioned already, SAHA is an approved drug. Other HDACs possibly would be less toxic or better tolerated, and we would suggest HDAC6 specific inhibitors would probably be the best. We are very interested in these two compounds right now that just came on the scene. These are the drugs of the decade, maybe. These are drugs that are absolutely fantastic for the treatment of multiple myeloma and hematological malignancies. They actually also work via hexim. I don't have time to go into, but their major effect, besides releasing BRD4 from chromatin, is that they vastly increase the synthesis of hexim by first releasing P-TEFb from the inactive. And they're soon to be approved for clinical use in humans. So basically, some sort of combination that you can see on this slide should be contemplated or will be contemplated for clinical trials, most likely in the monkeys. I would just like to say that other people have suggested that chromatin would be important to target, except we have recent finding that 5-azacitidine, which was supposed to just demethylate the DNA and therefore allow for better initiation of transcription, is also a very potent activator of P-TEFb. So I'm not so sure what people are looking when they're looking at azacitidine, whether they're actually looking at chromatin or whether they're actually looking at the P-TEFb cycle. And the others are obviously mentioned already: Braustatin, Prostatin, Ingenol, TNF for NF-kappa B and P-TEFb, and HMBA, SAHA, or others for the release of P-TEFb. So we believe that this could have an acronym: highly active anti-latency therapy, for us HALT. I'm not sure this is a good acronym. No, but the problem is that no good cell culture model exists for latency. That's the problem. No good small animal model exists, especially with problems with NOD background mice, which have activated the immune system. Probably the best would be to do all the trials in the SIV-infected rhesus macaques, which are very expensive, but probably it will have to be the basis for any clinical human trials. And then, obviously, for transcription interference and to increase levels of P-TEFb, some activator of NF-kappa B will have to be used. And for factor insufficiency, we'll have to increase levels of active P-TEFb or pan-HDAC inhibitor like SAHA. And with SAHA, I think that basically our results suggest that it's even being used not correctly, because the SAHA cycle is 6 hours. So we would actually contemplate a smaller dose more frequently in these individuals. Now, obviously, the problems are this, what I already mentioned: what combination of drugs. Dosing is going to be very difficult because of the cyclic nature of this, both NF-kappa B and P-TEFb. How good is going to be delivery of these agents to different organs is another problem. Duration will probably have to be for months to years. Side effects are not clear. I mean, probably rapidly dividing cells are going to suffer from this treatment. And how are we going to monitor this particular treatment is also a big problem. Now, just 1, 2, 3 slides. So basically, alternatives to activation therapy, which I presented to you in the preceding slides. There is an idea that starving the reservoir via blocking any cell activation might do the trick. I think that that's going to be probably not a great idea, especially since starvation for tumors has been so disappointing, and I don't see that if it's already factor insufficiency and transcription interference, this is just basically reinforcing those particular blocks to HIV replication. Should a marker for the reservoir be found, elimination via antibody toxin approaches could be contemplated, but I tell you the only marker that we have is the short transcripts, which are not on the surface. Gene therapy with specific custom-made nucleases, such as zinc or TALA nucleases, is an interesting idea. If you can target very conserved part of the virus that don't change, the zinc are probably not as good as the TALA, because the zinc requires a specific CpG sequences. TALA are actually more flexible in terms of recognition, and obviously gene therapy. So just to contrast, so the zinc finger nucleases or gene therapy, unfortunately. So zinc was already mentioned prior to this talk. They basically zinc fingers that target CpG sequences. So if you find enough of those in the virus, which is usually AT-rich, is unlikely. Now, TALA is a bit more interesting because you can modify the amino acids so they see any nucleotide sequence, And so basically this probably could be targeted a lot more precisely, but still, you have to make sure that you have covered all the variants of the virus or all pseudo-species or pseudo-species of the virus and you have to also be able to target the reservoir, which already would be difficult with drug, but maybe with gene therapy even more difficult. And then finally, if you really contemplate gene therapy, like the Berlin patient, except instead of CCR5 delta 32, you could actually argue that if you put, for example, SAM-HD1 into all cells, if you increase the expression of Tregs, for example, or RNSH2, or RIG-I, or innate sensors, or anything else that you think about that's tethering, for example, if you just increase the levels of that in the stem cell population, you would also be able to make cells less hospitable to virus, and you would actually make two populations, and the population with this particular restriction factors would presumably win out. But these are all gene therapy. I think it's very unlikely you'll find a specific promoter that you can target exogenously for any of these genes. So that's basically my presentation, and I'd like to thank the 3 Brazilian people in my lab who were extremely productive, who are now all back, or at least Tatiana is still in my lab, but Renato Aguiar and Luciana Costa and Tatiana Sampaio, who've done much of this work, and others, including these other people in the lab, and this was all done in collaboration with Paul Lucio, David Price, and to some extent Amilcar Tanuri in Rio de Janeiro. Thank you very much for your attention.